Hello and welcome to Physiology with Shakti, and in today's episode we're going to do with neural regulation of respiration. So if you haven't already watched the previous episode, please go do that because I had discussed a few important concepts about central pattern generators, about the inspiratory RAM signal, how it is acting, and the theories that are there behind it. So once you have a clear picture of that, you'll understand this topic a little better. So now we'll go to the neural regulation of respiration. So that central pattern generators we had mentioned earlier is how automatic pacemaker like activity is generated. Now that needs to be controlled. So there are two sets of ways you can control it. Neural control and through chemical control. So by the perception of composition, chemical composition in the body that regulates the respiration, and also by neural impulses. So how does these neural regulation take place? So that is what we need to understand in today's episode. When we talk about neural regulation, we need to understand where the various structures are with respect to your brainstem. So over here we got the pons, we got the medulla, and we got the spinal cord starting from the cervical segment starting from there. And above that we have the midbrain and the cortical portions above it. This is a part of the brainstem which is involved with regulation of respiration. And the pacemaker activity is predominantly present in this region in the medulla. We had seen it last time out. So in this medulla, in the center, we have a group of target region, and that is the nucleus tractus solitarius. So this receives inputs from different parts of the body to regulate things like blood pressure, respiration. It even takes impulses for taste sensations and takes it forward. So that is a integrating complex, the nucleus tractus solitarius. So when we take a look at the nucleus tractus solitarius, it is present dorsally in the midline, and just overlying those set of nucleus, we have another set of neurons which are present, which take up the dorsal group of nuclei. So on either side, this dorsal set of neurons. So they are the dorsal group of neurons. So they are usually termed as DRG. So DRG, the dorsal respiratory group of neurons. And apart from that, this is dorsal. Now ventrolaterally, we have the next set of neurons which are present. So this group is ventral and lateral. So ventrolaterally, we have another set of neurons. So this is the DRG, and over here we have the VRG, which are ventral respiratory group of neurons. We have the dorsal respiratory group of neurons, we have the ventral respiratory group of neurons. And this is where your pacemaker potentials are generated in respiration. With respect to respiration, we have seen it last time out. So that is your medulla, and these are the dorsal and the ventral group of neurons. And apart from that, in the pons, you have Pontine centers that further regulate this pacemaker activity. The central pattern generator activity is regulated by set of nuclei in the pons. So that is where we have one center over here called the apneustic center. This is the apneustic center, and we also have a pneumotaxic center. So this is a pneumotaxic center. This is a apneustic center. So we got the pneumotaxic center, apneustic center in the pons, which are involved in pontine regulation, and in the medulla we got the dorsal respiratory group as well as the ventral respiratory group. So these are ventrolateral and these are dorsal and medial. So that is the basic structure of the things that are involved over here. So these central pattern generators ultimately will produce your ramp signal. So this is finally produced by this, and that is regulated by your Pontine centers as well. So let's take a look at what one experiment done by Brainstem. So at a time when people didn't know what the brain centers were, where the centers for respiration were, this person called Brainstem, he tried out sectioning the brain in different parts to see where the center is. So let's try sectioning it here. This is the final outflow. When that final outflow was there was no respiration that was seen. Initially, without anything, the respiration would be like this. If you measure it on a spirometer, it's going to be like this. So this respiration will be absent because the final output is gone. Now let's take a section over here. Instead, we take a section over here somewhere. What is going to happen? That is where your main central pattern generators are present. So your respiration becomes very irregular. Sometimes it will be long, sometimes it will be short. It becomes very irregular. And that depends on where it is sectioned basically. More towards the bottom part would make it go more towards this. And the more above, the less influence on this. So it depends upon amount of destruction done to the medulla and these medullary regions like the dorsal respiratory group and the ventral respiratory group, which are involved in respiration. Now this person also tried to make a section over here. So what he saw was he got the normal sort of respiration. So a section above, including the cortical region or the midbrain, so those regions did not affect the normal respiration too much. Even in their absence, the person was still able to respire quite normally. When there was a transaction at this level, at the final outflow, that resulted in no respiratory signals. Pacemaker activity was not there. When the transaction was at the level of the medulla, because the central pattern generators are over there, it resulted in irregular bursts of activity. Now, the remaining things I'll come to a little later. So now we'll focus on this and see what happens over here. So we got the dorsal respiratory group as well as the ventral respiratory group. So I make this the dorsal respiratory group. So what this dorsal respiratory group does is, it results in this inspiratory ramp signal in passive phase. So imagine us breathing normally, tidal ventilation is taking place, and that inspiration is through your dorsal respiratory group of neurons. Now you know tidal expiration, eupneic expiration is passive. So what happens is simply switch off of the signal would result in the recoil and passive expiration. So dorsal respiratory group is involved with eupneic inspiration. On the other hand, eupneic expiration is passive and does not require all these neurons. Now when we talk about the ventral respiratory group, they are especially involved in overdrive mechanism. When we need to inspire more or expire more than normal, that is when your ventral respiratory group of neurons come into play. So this ventral respiratory group of neurons, we got the main core complex. There's a main core of this ventral respiratory group, which is formed by the Botzinger complex. So we got the Botzinger complex. So over here we have a Botzinger nucleus, and just over here there's another one which is pre-Botzinger. So we got two things, a Botzinger and a pre-Botzinger. So this together makes up a Botzinger complex. We got the Botzinger and we got the pre-Botzinger. Now this forms the major core of this ventral respiratory group. Now, apart from that, we have the medulla over here, and we know that in the medulla we got the vagal nucleus, and vagal nucleus runs throughout, but there's a vagal nucleus, parts of the vagal nucleus is there, nucleus ambiguous is there. Apart from that, we got the hypoglossal nucleus over here. So all these things are over here. So I just mentioned to you about nucleus ambiguous. So we got the nucleus ambiguous over here. So this is a nucleus ambiguous. We got the Botzinger complex, a pre-Botzinger complex. We got the nucleus ambiguous. So nucleus ambiguous gives supply to parts of your pharynx, larynx, all these regions. So this group of nuclei, so from the vagus, nucleus ambiguous, and parts of the hypoglossus, glossopharynx, all these together will represent this one area of the pharynx, larynx, and all that. So that is your nucleus ambiguous. This is nucleus ambiguous para. Next to nucleus ambiguous is nucleus paraambiguous. So over here we got the nucleus paraambiguous. So this is nucleus paraambiguous. So we got the nucleus ambiguous over here, nucleus paraambiguous. And what is there behind that goes around? That is retroambiguous. So we got the nucleus retroambiguous over here. Nucleus retroambiguous. So are the structures clear? We got the Botzinger complex, a pre-Botzinger complex. And then we got the nucleus ambiguous, nucleus paraambiguous, nucleus retroambiguous. This is your ventral respiratory group that I just mentioned, simply like this. So if you zoom into that, this is how it is. So we got the dorsal respiratory group and we got the ventral respiratory group comprising of the Botzinger complex, a pre-Botzinger complex, nucleus ambiguous, nucleus paraambiguous, nucleus retroambiguous. Now to understand how they function, what they are important for. Now we know that dorsal respiratory group is involved with inspiration in eupnea and expiration is a passive process. And when we come to ventral respiratory group, I had mentioned that it is involved in overdrive mechanisms when we need to inspire more than normal or expire more than normal. So in this scenario, your ventral respiratory group has different parts. Whatever I've shown in green is involved with inspiration and whatever is shown in blue is involved with expiration. So out of this, I had mentioned that this is the main core complex. We got the main core complex and they have a reciprocal feedback inhibition activity. So when pre-Botzinger is active, it will inhibit Botzinger. When Botzinger is active, it will inhibit pre-Botzinger. So basically when inspiration is happening, expiration should be stopped. When expiration is happening, inspiration is stopped because of this. And another thing you need to understand is this goes to the opposite side. Botzinger complex goes to the opposite side and from there it goes down and goes to the abdominal muscles and the intercostal muscles to result in expiration. Pre-Botzinger complex, that will go to the opposite side and it will go to parts of the diaphragm and the intercostal muscles and accessory muscles and result in inspiration. So that is what happens. We got the pre-Botzinger, nucleus ambiguous, nucleus paraambiguous, things in the center which are involved in inspiratory activity. So they go to the inspiratory muscle. And we got the Botzinger complex and the nucleus retroambiguous which go to the opposite side and go to the expiratory muscles. And at the same time, this Botzinger complex is going to inhibit the activity of inspiration and pre-Botzinger complex is going to inhibit the activity of expiration at the same time. The inhibition is also there. Apart from this, we got the other areas. I had mentioned about nucleus ambiguous, and I had mentioned about parts next to the hypoglossus, glossopharyngeal, and then we got the trigeminal and all that. So based on that, through cranial nerves, further control. For example, the anal nasi should act. Your mouth should open. So that's through trigeminal nerve. Anal nasi, so that should open up, flare up, and that acts through facial nerve. So through these different cranial nerves, they act on the same side to act on these accessory muscles as well. So goes to the opposite side and acts on the muscles that are involved in respiration, and for the accessory muscles, the cranial nerves go to the same side. So that is what happens over here. Now, when you talk about this core complex, the Botzinger is responsible for the main expiratory effort. So when we had talked about central pattern generator, we had mentioned about early inspiratory, inspiratory augmenting, late inspiratory, and expiratory decrementing, expiratory augmenting. So this expiratory decrementing and expiratory augmenting neurons are predominantly seen in the Botzinger complex and next to it. So that results in the expiratory activity, and further modulation can be done by your nucleus retroambiguous. In a similar fashion, the major inspiratory efforts are through the pre-Botzinger complex, and further modulation can be done with the help of nucleus parambiguous. And I already mentioned how nucleus ambiguous goes to parts of the pharynx and larynx. So inspiration, pre-Botzinger complex, expiration, Botzinger complex. Further modulation of inspiration, nucleus parambiguous. Further modulation of expiration, nucleus retroambiguous. And nucleus ambiguous for inspiratory efforts need to abduct the parts of the laryngeal muscles, and pharynx should dilate, and that sort of response is through nucleus ambiguous. So this is what happens in the medulla, and when there's a lesion over here, that results in irregularity, and that depends on how much of these structures are damaged. If there is complete damage of all this, it's going to be like this. But since there is one transaction at one part and certain parts are still remaining, that is why it is coming as an irregular pattern. Is this clear? That is your central pattern generators. And what happens in the medulla. Now, apart from this, we got a Pontine control. Above this, the Pontine control. So we got two things, apneustic center and the pneumotaxic center. So I'll just draw this RAM signal again over here. So this is your RAM signal, inspiratory RAM signal. So normally this takes around 2 seconds time, and expiration goes about for 3 second time. So this is a normal scenario that we see. Now this apneustic center, what it does is it acts on these central pattern generators in order to prolong the activity. So instead of this, if you want this activity to be prolonged further, that is when your apneustic center acts. So apneustic center acts on the central pattern generators to prolong respiration. So when this is prolonged, more inspiratory effort, more recruitment of inspiratory muscles, and that results in increase in the depth of respiration. So this is what apneustic center does. Now, this sort of prolongation is inhibited by your pneumotaxic center. So what pneumotaxic center does is it inhibits your apneustic center. So ultimately, it decides when to stop the signal. So when you stop the signal, it falls rapidly at that point. So normally it's set to this level. If it realizes that, okay, you need more breath, you need more air coming in, what is done is that it relieves the inhibition so that apneustic center can now act freely to prolong respiration. In case you want faster breaths, you don't want deep breaths, you want shallow rapid breaths, it is going to stop much earlier, and instead of stopping here, it will stop over here. So that is your pneumotaxic center. it controls how acoustic center acts on the central pattern generators. So a cutoff point. So normally it is 2 seconds I had mentioned. So this comes to 2 seconds. Instead, it can make it go as short as 0.5 seconds to as long as 5 seconds. So this is the range to which your pneumotaxic center can act by modulating your acoustic center. Normal breath takes, inspiratory effort takes, the RAM signal takes around 2 seconds. It can cut short it to as low as 0.5 seconds. And it can prolong it to as high as 5 seconds. So this is a normal range to which it can work. So 0.5 seconds means the person ends up breathing 30 to 40 times a minute. Normal breath is 12 to, when it is 2 seconds, normally it comes to 12 to 16 times per minute breath. But when it's cut short and the inspiratory effort is shorter, and when it is 0.5 it can go up to 30 to 40 breaths per minute. And in a similar way, if it is prolonged to up to 5 seconds like this, then that breath, which was 12 to 16 breaths per minute, can go as low as 3 to 4 breaths per minute. 3 to 4 breaths per minute. So that is the activity of your Pontine regulators. The pneumotaxic center inhibits your acoustic center. Acoustic center prolongs this activity. So there is another thing. Apart from your pneumotaxic center, we got the vagus also acting over here. So we'll come to that later. So right now we'll just try to understand what happens. So I said that over here, if a transaction is done, it is above the pneumotaxic center and no other regulation is affected. Thus, it kind of remains normal. The only thing is all these emotional things might not affect it, that's it. So the pacemaker potential remains normal and normal respiration takes place. But if we now go on to make a lesion over there, then pneumotaxic center is gone. So what is going to happen? Pneumotaxic center's influence is gone, which means acoustic center is free to act. So it will keep acting. It keeps acting means depth of respiration increases. So what happens here? From this, the depth of respiration has increased. You see that? From here, the depth of inspiration has increased because of a lesion over here. Understood? Yes. So now I had also mentioned about the role of vagus. So vagus finally goes from different parts and reaches here nucleus tractus solitarius. So the vagus is coming. It reaches over here and from here it goes to different parts. The regulation goes to different parts. So one way it does is by inhibition of your acoustic center. So just like your pneumotaxic center, there is an inhibition of your acoustic center. So I'll draw another set of scenario over here where vagus is cut in each of these. Now at this point, if vagus is also cut, doesn't make a difference. It's still not going to result in anything. Final output will still not be there. You cut a vagus over here along with cutting it at the medulla. This is still going to be regular because this is the main central pattern generators. So these things are not going to change. The only change is going to be above this. Now let's see what happens. We have made a transaction over here. Pneumotaxic center is paired. So that is normally inhibiting acoustic center. But now we're cutting vagus. So normally it's like this, but when we cut the vagus, what we see is the depth of respiration is increased. It's not difficult to understand. It has a similar role as a pneumotaxic center to switch it off earlier, to inhibit it. So when that effect of vagus is gone, there is a similar scenario over here. So there are two things that you need to understand. One is the pneumotaxic center, other is the vagus. Two things are involved in inhibiting and keeping your apneustic center in place so that it doesn't keep prolonging. Two things are required. If any one of them is gone, the other one takes over. That is why over here, pneumotaxic center is transected over here, but your vagus is still keeping it check. Over here, your vagus is cut, pneumotaxic center is intact, and that is why your pneumotaxic center is still keeping this in check. So over here, pneumotaxic center is cut, but vagus is keeping it in check. Here, your vagus is cut, but pneumotaxic center is keeping it in check. So you see how these two are similar? At least one of them should be there. What if both of them are not there? So let's take a transaction at this point. You cut the pneumotaxic center, and at the same time, you're going to cut the vagus as well. So we cut both the vagus and pneumotaxic center. So there is literally no inhibition on the apneustic center. So it keeps prolonging. So that is what is called the apneustic type of breathing. Keep going like this, and ultimately it stops at one point. That is the apneustic type of breathing. Apnea refers to temporary cessation of breathing. A temporary cessation of breathing. So even if it is inspiration, there is a temporary cessation of breathing over here. And there is a gasp followed by expiration and then again inspiratory gasp. So that sort of breathing will be seen in this type of lesion. So now the question is, if it is going to prolong it, why doesn't it keep happening? That is when your chemical regulators come into place, not only neural regulation. There will be changes in the oxygen, carbon dioxide, pH because of prolonged inspiration and cessation of breathing. So because of those changes, ultimately at one point, your central and peripheral chemoreceptors will come into picture. So your central chemoreceptor is very powerful. They will override this and make it stop. So that is your apneustic type of breathing. So this is very simple. It looks very complicated, but you just have to understand these areas. This is normal. And I focused on this because questions are coming from it. They'll put an image-based question for this. They might put the question and ask lesion here will result in what? Or they might ask about the apneustic type of breathing. Lesion of this and this will result in this effect. So questions from this will be coming. Questions from this will be coming. And questions on what Pontine centers have effect on your RAM signal. That is another set of questions that come over here. So that sums up your neural regulation. I hope this is clear. So if you understand central pattern generators and related to what I taught over here, then it will be simple to understand. In the next episode, we'll go to chemical regulation of respiration. So thank you. So if you found the channel to be useful and informative, please do share it, let other people know, and let them also understand the topic just as much as you did. Thank you.